we have seen a lower density of animals throughout this dive, and
Got a nose.
that might be a possible explanation.
10 meters 040.
And
Yeah. I'll put it in.
Copy. Just to help them out a little.
I'm going to pop up just a little bit and try and frame all of these guys.
Sure thing.
I'm not sure that other one's going to fit, but it's a nice overall shot. Characterizes this area. Yeah, right in there. That's good.
so the closest we had for this was in the Hurl guide, but the Balsaroides daphnia, but I believe this is something different. And I think that's we identified as, I believe, Relactus is the genus. Is that right? Scott had provided a tentative ID for this group.
Group photo.
It's hard to place the outside of your toe. We'll get there. Okay. There it is. Found my toe
Some
hold.
more light for you.
Thanks. And you can come on in, video.
Great. I'm going to come in for focus and establish this.
Nice.
Let me get the whole organism.
Excellent, thank you.
Can we tilt down just a scooch, please?
Good?
Yeah, that's perfect. Perfect right there. Do we we're not doing a ship move, right? We're stationary?
We're okay.
Stationary, yep.
Thank you.
We're doing a 10-meter move.
D2 is fine for now. We're only 10 meters for us.
Yeah.
All right. Let me go ahead and see if we can get a little bit of this. The one we had the other day had a little more current, so they were a bit more active.
That nab we just inched in just so I can get, so he can not feel the tether pull as much, yeah.
There we go.
Hopefully that should be done soon.
Is there any way to get those particles out of there, or it won't look as nice on the other guy? I kicked up a lot when I sat down.
Yeah.
Excellent, beautiful imagery. Thank you.
We'll let Art take each one of those out separately in post.
For the highlight.
So yeah, this is a very, very large species. For an anemone, yeah, those laser pointers are 10 centimeters apart. So that is over 20 centimeters across. Pretty large for an anemone.
Those little manip cams do an amazing job picking up color.
All right, let's see that come out a little bit.
Great.
Let me get rid of that bright spot. Feels like we've got this.
All right.
Coming out, adjusting our levels. Want that one in the background at all? Is there any interest in that, or have you guys imaged that this morning?
I don't know.
See if science says anything about it.
I think we should characterize this here.
Nice little cove.
Back out before you...
Weird discoloration in the sediment down below.
Yeah, let's take a look at
Intentionally
think,
use the
the
word wet to describe it.
sponge to our left looks like Lafroyella, I believe. But yeah, maybe we can take a quick snap zoom.
Yeah, you snap in from here.
Let's try it.
There's a lot of dust between us and there, too.
Yeah.
Oh, that's a pretty good characterization right there.
So Daniel, I don't think this is Lefroyella. It doesn't have the commensal anemones on it, but it is a euretid. It's one of the hard sponges, and that's about as good as we can get on this plate one. And then the one to the upper right is the Faria in the family Fariidae.
Back in there.
Video you
Have you folks been on
partial on Sirius briefly,
Yeah.
just to get that kind of cave shot. That's a little unique.
Yeah, so above
Hopefully
it,
we'll
that's the
stabilize
Faria
that.
near
Of course,
Oka
we'll
erecta.
stabilize at the top.
Once we start bouncing, come wide.
And again, so
Yeah, just let me know if you see it stable again.
these are some very
Hey, pilot, why don't you go
unusual shapes in sponges that you see for a lot of these glass sponges, and that's because they're very different and in a completely different group than those sponges that you typically see in shallow waters.
I think we'll be able to go back down. I just want to,
Double check.
yeah, do that whole dance again.
All right.
Okay, good. My sonar targets are getting further away from me. That makes me more comfortable.
We really were down in a little cave thing, huh?
Yeah.
It's cool. Not in, but looking into.
So if you're, since the ship's not moving, if you'd like to go back down and catch one of those, you can.
It's up to science. Was there anything else in that little overhang you wanted to see, science? Or is that, or did we get a characterization?
Yeah, I think we're good. Thank you.
All right.
Yeah, we have about an hour left on this dive, and it looks like we're just under 100 meters away from the summit. As Jonathan had pointed out, we are getting into levels where the oxygen level is dropping quite low. It's not the true oxygen minimum zone, which is typically defined as places where you have oxygen content below 1.0 milligrams per liter.
It's not landing on the hot part.
Daniel, would it be possible for them to put the Sirius view on camera 2 just so that the students can see the vehicle a little bit larger?
Roger that.
So video, we've had a request from Shore-Based Science to get a full screen view from Sirius on camera 2.
Roger, stream 2 is now Sirius. Watch like video, I could probably do a push on there if co-pilots are okay with it.
Stand by one for me. Yeah, I'm okay with that briefly.
Okay, coming in 3.
Can we get a zoom on that coral that's just right at the bottom of the picture right now?
And coming out on Sirius.
The way they engineer these things is that they have a vehicle that's tethered to the ship.
Awful one.
So as the ship moves, that vehicle is moving.
You all right?
That's this one.
Okay. I thought you said
And then
there's
coming
some
out of
videos
that first vehicle,
Video, I think.
So there's
they wanna
also,
come in this.
yes, I believe so, and that's a Chrysogorgid coral, and it's got this commensal squat lobster in the family Chirostelidae
It's floating around.
that Mary had identified as Europtychus.
So Chris, I can hear you over the conference line. Can you
And then another guy doing the camera and the robot arm.
videographer doing the cameras, pilot doing around, co-pilot doing the upper vehicle.
Get closer.
And then
So.
there's a navigator taking
So, Chris, I can hear you over the conference line. Can you mute the conference call in the ECC, please?
Coming down for you too.
So there we have.
Just trying to make small movements so we don't get an isolation.
Yeah, there's this particular coral always has earlier, and because every time we look at it, both in the Atlantic Ocean as well as East Coast,
There's no way to get
we find this same shrimp.
Why is it that?
So,
Perfect, excellent. So yeah, that's a chrysogorgid coral, and with that commensal squat lobster again. So yeah, thank you.
Gotta get the whole thing here before we have to do it.
Pilot, when you come out, it looks like there might be another overhang above you. So
Copy that.
Can we wag right and see if we can get a two-shot before we leave? This on the left and the other organism up and to the right looks like crinoid or something like that.
Yeah. Do what you gotta do with Zoom to make it happen.
Just 5 of this. Got it. Thank you.
Yeah.
Coming all the way out.
Roger.
Full wide. Huh. This one coming up looks cool in Zeus, but it's hard to tell.
yeah, it's. I don't have any depth perception there. I think looking at your
Wow.
mini Zeus, you might want to back out. Yeah, you're good. Yeah, that's an overhang for sure.
Somebody order a sponge.
A lot of it.
Somebody ordered 7, 8, 9 sponges.
Chris Kelly will be happy.
Ooh, is that one of the bivalves?
Ooh.
There's 2 of them, it looks like. 2 or 3.
Like the ones we saw on the bottom.
Yeah,
Yeah.
looks like where that might have come from. You want to come in partial right here?
Partial eye.
I'm just going to try to get a little closer.
And
too.
holding.
Thanks. You're going to get some extra light. I'm coming in a little bit, but it is overhung, so not too far.
Clear for a bit. Coming up on Sirius.
Copy. I'll hold there. Is that a bivalve? Is that a sponge? Looks like a sponge, I guess, huh?
Can't
Jump
tell.
the gun. Look at that ring in the very top. Did you see that?
Just one frame right there.
Yeah. It's kind of interesting, isn't it?
I wonder what changed that gave us all this life in the last 20 meters.
Or getting towards the pinnacle, maybe?
O2,
I guess we still got a ways.
temperature.
O2 went down, I think.
Yeah,
Yes.
the DO was reducing.
Yeah, O2
It's
drops
like
at some point.
1 right now, right?
Yeah.
It's pretty deadly.
So yeah, we just got a little quiet there. We're talking here with Brian about plans for tomorrow, and we will have a call after this dive, so about an hour from now when we come up.
Just scanning around and looking at the creatures that we can see.
So yeah, now,
Reflection.
there's an unbranched wire or bamboo coral. It's in the genus Lepidisis as for now. Then we have Furia near Oka erecta. We have also couple of crinoids, nice community, and you can see there like the dead sponge material to the base,
We're eating a little glare.
and then you can see the live sponge that has a different
So
color.
It might be just this
And
monitor too.
on the Sirius overhead, you can actually see that we're coming up on a nice cluster of animals.
Alright, I'm gonna push up here. We got a sort of nice shot of everything. Slowly rise and follow the biology. An interesting geological feature.
And there's one of these large anemones, that left one to that orange one, that's Exosalactus, is one of the larger ones, and it looks like next to it, the smaller one is a Venus trap, and then we're coming up on another Chrysogorgid, that's going to be in a genus Iridogorgia. And yeah, it's a beautiful view here as we're getting to the top of this pinnacle. We're just 1,200 meters right now. So we see a significant increase in the fauna and the density of animals. A lot of sponges and corals. So a lot of these Furia near Oca erecta, and then several of these Atlanticella sponges, several Chrysogorgids. So yeah, beautiful view here of a dense aggregation, and on the Sirius overhead, you can really see that the density of animals is starting to pick up.
Oh, there's a big basket star.
Yeah, let's take a look at him there. That's one of the most.
There's a couple of. Oh, that's just.
This is one of the densest aggregations of organisms we've seen on the entire dive. Does anybody care to speculate why?
So we are coming up onto the top of this feature, and that's typical. Yeah, is when you see
I think that was
pinnacles and seamounts in general, or rocks is on the upper flanks and
we
summits.
them on the basket. I was waiting for you to.
And that's just because you have
for you to
kind of water
out seats.
moving in a circular motion
when you're
around
ready.
this place,
Sounds
and
like you are.
so animals that filter
When they
feed or
rise up.
feed on suspension aggregate on these places where you have higher current flow. And we're going to take a look at this basket star and the family Gorgonocephalidae. One of the most spectacular animals you'll see. So these are related, so they're the same group as urchins, sea stars, or sea cucumbers. They're characterized by having 5-sided symmetry. And yeah, these are absolutely beautiful animals.
Excellent.
Thanks,
I'll
Roland.
just drift around it and I'll follow focus.
Yeah, and back to the question here about the aggregation of animals. So yeah, I did update the position, and then we are getting to the upper flank, so that almost top of the pinnacle. So what happens is actually, they've been called Taylor columns. So these circulation features that kind of are on top and around pinnacles and seamounts, and these extending for long vertical distances above it, and that kind of traps food in the area.
Pulling out
of course, that provides prime habitat for those organisms that feed on tiny particles that are in the water column, things like plankton. But it's not only high current that these organisms like. They also need stable environments, so that's also something that we've seen, that there's a kind of a delicate interplay between both high current flow or dependable current flow, I should say, and then also stable environments, so in places where the substrate is kind of more gravelly or loose rocks, and you might have slides, then that does not provide for a dependable long-term habitat. And then from the few dives what we've done in high current environments, there's also a thing as too much flow, and so if the currents are too strong, then it basically tumbles over everything, and also doesn't allow for things to settle. So a lot of these organisms have planktonic larvae, and they need to settle onto the substrate, so they need a place that they can settle on. Yeah, absolutely spectacular view now of just the extreme high density. Yeah, maybe we can take a look at that crab real quick.
Okay. Get the crab for you.
Did you guys know that this class is going to come here at this moment and set up this dive where there's tons of animals here, right when they're in the ECC?
That is excellent. I'm glad.
Yeah, Chris, we actually planned that. Speaking of dive planning.
Yeah.
Tilt up toward that guy.
It's kind of like those jet fighters going on top of the Super Bowl before kickoff, yeah. That kind of coordination.
Okay. There's just a lot of glare there, huh, Roland? Ready? Yep.
Yeah, that's the same one. I thought he looked different. He's lost one of his claws, though.
He's pushing tight to his face.
Okay.
Oh, okay.
Settled in when you are rolling. Set full zoom.
That is now max zoom.
So our current depth is 1,096 meters. We're just less than 40 meters in vertical distance away from the very summit of this feature.
Partial.
And yeah, the density of animals has kept increasing as we move towards the upper flanks and towards the summit here. On the left is another one of these glass sponges that Chris mentioned. So glass sponges is a very large group of sponges that's characteristic of the deep sea. So they have a very kind of spiky surface. When you touch them, they're not smooth to the touch. And that's because their skeleton is made out of these siliceous skeletal elements that have pointed features.
Awesome.
And they are very common in the deep sea and then very rare in shallow waters. Only a few species of glass sponges that are found in shallow waters, and certainly no shallow water glass sponges in Hawaii.
I know, this is steep.
Yeah.
And yeah, that's a nice view that you see both live and dead sponge material. You can see the difference in coloration. But an important feature is why we're looking at these ecosystems or why we're studying these deep aggregations of corals and sponges, is that these animals provide habitat for a lot of other invertebrates. So oftentimes you see little brittle stars or little shrimps and crabs and all kinds of things living amongst the corals. And even after the sponges die, that structure is still there for other animals to take shelter in.
Maybe just a stalk.
Yeah, it might just be a dead stock. You can zoom from here. Just get a quick shot. If they want to stay longer, we will. How much time is left? 40 minutes?
Oh, excellent.
Yeah, about 40, 45 minutes.
How long do we have until we're there?
If we start moving right now?
No, I just mean,
Or
what
distance?
distance, yeah. Just, yeah, missing head.
So D2's about...
Missing head. Caliphacus with a missing head or something.
You just keep drifting up.
And there you can see there's a little shrimp, and this is what I was talking about all kinds of other
so
things
about 100
living.
meters away. D2's about 80, 85.
Very nicely done.
Yeah, and little hydroids as well.
There's another lobster, I believe, over to the right.
Okay. Those are some cool looking guys. Go back in real quick.
In.
You see what I'm talking about?
There's a little crab, and that's the first time we've seen those large polyps there. Those are solitary hydroids,
Sorry, I was just
and those are
syncing
going to be in the
up.
family Corimorphidae.
Hydroid top right.
And so with corals and hydroids, you have some animals that are solitary, meaning that a single animal consists of a single polyp. And then you have others that are colonial, where a single animal consists of a multitude of polyps all stuck together. And an example of that is actually the animal that we're imaging right now, that yellow one to the bottom. So you have different forms. Atlanticellus. Yeah, these are very beautiful. And see some shrimps in there. You see some crabs in there. That might be a Munidopsis. That white crab on top of the sponge to the left, that's Furriania erecta.
White crab. Oh, there he is.
And yeah, just a beautiful aggregation of animals,
To the left.
all kinds of different
What
groups.
came to the view?
Shrimp top left.
Oh yeah, I saw that. A fair amount of current, I guess. I don't know. I'm just... Sorry, just readjusting here. Floating away a bit.
Video's clear.
Can we take a look at those small polyps on the rock to the upper right?
Yeah.
I didn't know what those are.
Max zoom?
That's Max?
Yes, sir.
Alright, I'll come in.
Following focus. Unless you want me to pull wide.
That's alright. I'll just come in here.
Yeah, so I don't know if Tina or Scott or Chris have a suggestion for those tiny.
Okay. Hold up
There is
here.
a
Getting pretty close.
Corallimorpharian that has kind of those distinct differences in colorations between the tip of the tentacles, but it's hard to say from this view. And Tina agrees with me, and
Sorry,
so
Rob.
yeah, the
I just
species I was thinking
trying to get
of
close,
is
but I've got my eyebrow right on a rock that I'm trying not to bump.
Corallimorphus pilatus.
Right near a bunch of sponge, so I'm not trying to bump.
The upper right had the best image of those guys.
Copy that. Going for it.
But typically
Tilt
you
up
see
now, help me
those
keep my eyebrows
kind of
up.
isolated. It's just one animal here or there, not a
I
big
keep my eyebrow out of there.
aggregation like this.
look like many anemones. The
Can't
video.
tell why I'm dropping so much. Am I feeling the ship heave at all? I don't think so. I'm not really stretched out.
I think video's got all we need. Science
okay.
is clear.
Thanks.
You did just get a rock.
Yeah, very.
But
And as soon as we zoom out, it gives you a bit more appreciation about how much life there is. And yeah, I'd posed this question earlier and made a statement about,
Definitely.
yeah, for a long time
Got some
people
oscillation
referred
going.
to the
Might be
deep
my fault.
ocean as the azoic zone, so which literally means the
and
zone
away.
that's void of life. So
Let's get to
people
the top
thought
of
for a
the
long
pinnacle.
time that
All right. What type of
at deep
jump
and
was
dark
it?
depths we
Let me-
don't
let
have
me get up
life,
a little
and
farther and let-
I hope to convince
let me
you
stretch out.
with these images that there's
idea.
actually a lot of life in many of these places.
Call it in.
They're important habitat
right?
for
18. Oh, I am pushed
a
out
lot
pretty
of
good, huh?
animals,
To some degree.
and actually these
yeah, that might have been
deeper
waves.
ecosystems are actually the most abundant ecosystems on the planet. If you look at by depth,
It's very
most
small
of
normally,
the
but
surface
when
of the
you zoom
Earth
in-
is in deep waters. So we don't often think of ourselves as being the strange ones, but yeah, if you take the planet as a whole, this is what most of the planet it looks like. And yeah, beautiful imagery here. Current depth is 1,190 meters.
Science, do we want to take stream 2 back to the multi-view? Currently it's on Sirius.
Let's keep Sirius, I think, if that's all right. Yeah, we can keep it on the Sirius.
Copy that.
Yeah, and Les just chimed in there. Yeah, I did not know exactly what this was. The only thing that made me lean towards Corallimorpharians was those white tips of tentacles, because we do have one species that has that characteristic
yeah, am I
coloration.
pretty stretched out here? I think that this is maybe a good spot. So I might wanna
But
look around
I have
here.
also never seen a dense aggregation like that. Typically,
know
you
if
see
I
them
have
just
to go
isolated.
there.
You want to do
Can
15 or 20? What, more than that?
yeah, I'm pretty stretched out, right? Yeah, we could do 20.
Yeah. We could do 25.
So Daniel, I don't think I've ever seen a community like this in any place we dove on before, where it's dominated by sponges, and the actual sponges that are in this community are very different than ones that, such as the Poliopogon, which is where you more commonly see, or the type of sponges that you more commonly see at these depths in large aggregations. So this is really unusual.
Yeah, Chris, and then, well, the feature itself is extremely unusual. Yeah, when I woke up this morning and saw the mapping of the area, so I'd never seen a pinnacle like that steep. So this is on one side of the pinnacle. It's basically 500 meters of sheer vertical wall down, and it's quite spectacular.
This is about stretched out here, so this is about as close as I can get at the moment. If you want to take any zooms here, you can catch something along this wall.
Would you like a partial to drift around with?
Yeah, sure. Thanks.
Partial.
Awesome.
Coral behind that stalk.
There's a very massive Seiko Calyx sponge.
Coral.
We had
Oh, I
imaged
see.
a small one earlier in the
Can you
dive, but
bring
this
that
is a
out?
massive.
I'll get the white out of there.
And we see a chrysogorget behind it, and actually there on the stalk of the sponge, those are tiny barnacles on it. Barnacles, so the scientific name there for them is Cirripedia, which literally means curled feet, and you can see that those tiny projections are actually the
little,
feet that they stick out into the water column, and they feed that way. They have little mucus pouches that they attach to the feet that they use to capture
Hold
food.
on real quick.
And
tip there. One of them
so
is open.
barnacles are kind of upside
Barnacle
down. They're
open.
glued
See it?
to the substrate with their forehead, and then they use their feet to feed.
You see the open one, watch lead on the very end there?
They're a very interesting animal. So think about yourself
Oh,
just
you can't hear me.
doing a permanent
You
headstand.
see the open barnacle on the tip?
All its little
Think of
tentacles
yourself,
sticking
yeah, as
out.
doing an open handstand and using your feet to eat.
Okay. Can come out to a partial unless you want to keep this for a while video.
And another interesting thing is, so like, yeah, they're really small, but barnacles, they have these plates around them. So for a long time, people thought that this group of
Let's get a good
animals
shot of
was
one of those. Let's see if there's some in the foreground.
closely
Wanna come
related
partial real
to
quick? See if one that's a little better
mollusks
lit, a little
because of
closer.
these kind of shell-like. It was not until later that the larvae of these animals were seen, and you can see clear segmentations which put these things actually more closely related to shrimps and crabs. So they have these segments.
Completely covered with them.
Okay. Come on in.
And
we're
yeah,
gonna
so this
keep
is
coming
at
right,
Feria near
down
Oka
closer.
erecta. And there's more of that. Yeah, very unusual.
It's completely covering this rock structure.
Yeah.
All right. If you've got any more, zoom.
Max zoom.
Max zoom. Okay.
Yeah, so this
I've
is
got more tether. Do you want me to try to get a little closer real quick?
Yeah.
All right. Come on out, just so I can jump in.
yeah, this is puzzling a few of us. Yeah, I've never seen that aggregation
It's gonna be
of
maybe a
if
little
it is
bumpy. I'll just
Corallimorpharians.
jump in here.
It's absolutely spectacular.
Very large sponge.
Real treat, and I'm glad that some of our school groups were able to join us for this
Not to bump it.
absolutely stunning scenery. We are 3/4 of a mile deep, and just incredible densities.
The slope is a little.
So Bruce had asked earlier in the dive how did reefs look like in shallow water. So this whole feature actually gets to within 80
All right.
feet of the surface of the
This is
water.
comfortable distance in terms of the stuff on the walls
Up there
sticking
where
out.
we dove this summer, the whole substrate is carpeted by this algae. You almost have no corals and no sponges, so it's all dominated by an algae in the genus Microdictyon. And so down here, close to a mile deeper, it's a completely different community and a lot more diversity.
Flights that last move decided.
Back to.
Copy that. There's a little tube for you.
Let me know how you're doing.
And
I'll
that
fire
looks
up
like
and
a
jump off when you're
polychaete
ready.
worm tube.
Let's get this guy. Right here.
They're to the left of the screen.
I'll hold this until you're ready, co-pilot.
I think we're about ready.
Okay.
And
All right, video. I need to come out.
Pulling out.
there's
Hang
a
on. Say again. Watch lead. Okay. Taking off.
Just come up and
Up, 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 up.
up.
full line.
Thank you, Roland. Up and back.
So there's a yellow coral. Yeah, well, I guess we're on the move, but we'll try to keep our eye out for another one that we can get a zoom in.
Oh, sorry, watch lead. I heard you say something yellow, but prior, but I thought you were talking to somebody else maybe, because when I asked you didn't.
Yeah, and sorry, I had the mic muted, but
Oh, gotcha.
I'll keep my eyes open.
Okay. Yeah, we just trying to get you this pinnacle, so we're trying to keep the moves.
Perfect. Yeah, no, and that's the priority we have.
We've got 25 minutes and still a pretty good distance to go, so we're not trying to. We don't want to be unsafe, but we don't want to like sit around one spot too long.
Roger that. Yeah, thank you.
Is that a loose rock inside that sediment there on the left?
Not an ideal time to grab a loose rock, I don't think.
Fish also going to
Fish is a good thing to get. Okay.
It's another one of this
Zoom fish.
Rhodonirogorgia. Oh, and that's our Mercurid, and that's in the genus Caraphenoides. Those are rat tails, commonly known also as grenadiers. One of the more common families of fishes in the deep sea.
So, Daniel, I'm pretty sure this is Caraphenoides longiphilis. It's got a really long pelvic extension of the rays. So you'll probably find that in the gallery, but it's very distinctive.
Yeah, thank you, Chris, and I totally agree. That's exactly what it is.
We're going to continue on. Oh, there's another one. Snap into that fish in the background just in case it's something different. It is something different.
Yeah, so that I believe. Ooh.
Swimming away from us. Can you do it, video?
And Chris, I think that's also something that we imaged last year, but he's moving away from us. That looked like
got a bunch of rocks between me and it. I can't chase it necessarily. All right, I gotta come up. Thanks,
we need to see more of them besides his rear end.
Yeah.
He's down in a crack there.
So this thing is in the family Ophidiidae for sure, but I couldn't tell which.
Okay, we can snap a top-down view if I can get it. He didn't swim under a crack.
I think he kept going around the corner.
Okay.
Yeah.
Corner, you can't proceed.
Yep. If only we had fish-sized ROVs.
Is that a cup coral on the right?
So our depth is now 1,167. Looks like an anemone. That's, I think, a Venus flytrap.
Time for snap?
Yeah, but
Coming up with you.
Thanks.
yeah, so that's going to be in the genus Philiactis, and those are in the family Hormathiidae, and you have these distinctive bumps on the stalk of the anemone. But yeah, Chris Kelly pointed out that this is an extremely unusual community, and he would know. He's done probably more dives than anyone here in Hawaii on these deeper reefs. Yeah, this is unusual and a real treat to be part of imaging this dive that in a community that is distinctly different, and just the topography also on very peculiar and unusual. It's this enormous pinnacle that on one side has about a 500 meter drop. And to the left is a pink coral, also characteristic group of this here in the deep sea. But yeah, this is definitely what we would consider high density.
Snap on pink.
Yep. Sorry, go ahead.
Yeah, so that's going to be in the genus Hemicorallium. Oh, and so I believe that yellow coral in the background, that's the one that they would like to get.
All right, I'm going to track up to there, Roland.
And so I think that, yeah, it's a Acanthogorgia.
Pushing.
Okay. Trying to hold it for you.
A lot of very happy school kids just left the ECC, totally mesmerized in what you guys are showing us.
As I said, we timed that like the Jets before the Super Bowl.
Thanks. Did you get what you needed for that watch lead, or do you need more?
That's good, thank you.
All right, you're welcome.
Yeah, I think you created some new addicts. Some of them are saying that they don't care. They're going home tonight and they're going to watch some more of it, even if it's the after feed.
Yeah, thanks, Les. So yeah, he identified a Paramaricia. So that's a Plexaurid.
Look at that white coral between the top sponges.
White coral between the top
Should be
sponges.
a skeleton.
Yeah.
It's going behind that sponge.
Oh, I see it.
Yeah, but it's...
Okay, you can.
And
snap
there's
it.
another one of these crown
Oh, you're looking at the yellow one, right?
hydroids, the
Is that the one you're
Hydromedusa.
No, but this is a
Yeah,
good size.
so that's, I believe, and that's, yep. So at the base is another one of this Venus flytrap, or Hormathiid anemones in the genus Philiactis. There's a squat lobster there in the back. And
what was
that's
the white coral you're looking for?
It was up behind that
no,
sponge.
no, that's the one.
Okay. Oh, all right. Actually, got it. Great.
So yeah, we're
Moving onward.
very close to the highest point here. Our depth is 1,158 meters, and yeah, the
Jump on ahead.
the shallowest is just
We've gotten a
7
couple of
meters,
those
so
earlier.
shallower than us. So yeah, we are reaching the summit here.
Is that a spiral coral?
Actually, can we take a quick look at that brown animal to our bottom?
Yep.
I don't know if that's
You can snap in, Roland. Thank you, sir.
oh yeah. Yeah, so that's the Rodan iridogorgia, I believe, and a young one.
I'm backing away, Roland. Sorry.
Difficult to image. That background is as busy as the foreground.
Yeah. Okay.
See how much time we have left.
Did you get a good look at that watch lead?
Yes, thank you. We should have 21 minutes remaining on the dive.
Ship stopped.
So
Yeah, it's been stopped. Are you ready for another move?
Let's make sure that these. You don't want to pick any rocks from here, do you? Watch lead.
So yeah, we were just discussing this. So
Want to make sure before I grab.
Nav, do you know how far away we are from the summit?
So we're about 60 meters.
Yeah, so it's probably going to be one over the other.
One rock
Well,
in
what do you-
hand and
what do you
one
think,
in the
Dan?
bush.
Yeah, I think I would push on to the summit because we might have a flat surface up there, yeah, because if we stop here, that's going to be it.
Nav, you want to get a
Okay.
30-meter
Pinnacle it is.
Yeah, keep it at 20 degrees.
Just because of how unusual that community was, I assume that we'll see something.
30 meters at 025.
Good copy.
Are you guys still close to that Chrysogorgid?
Which is that the spiral one?
Yeah, the one that should be right beyond, underneath us. Yeah.
Are you looking to collect one more biological, or do you have them all? We were a little distracted with the class here.
Well, so yeah,
I
we.
think you've only got the one biological, right?
Yeah, so we still have one biological, one rock. The question was, so we have about 20 minutes remaining of the dive, so if we collect samples, we'll not make it to the summit. So it's either we collect, and then that's going to be probably it for the dive, or we carry on to the summit. And I'll leave, I'll put that up for discussion. But we are still about 60 meters from the summit.
If you, actually, if you decide soon, we just dialed in a ship move that will have to stop if you decide to stay.
Good question. At least we have one vote here to collect. That's Brian.
Yeah.
Les would probably think it'd be good to collect a rock and a biological as a way to spend the rest of the dive.
Copy that, Chris. Thanks a lot. So I think we need to make that final decision pretty soon, though, if we're going to do it.
Well, so let's stop that ship move. Sorry about that, Nav. Yeah, sorry about that, Nav and pilot.
You didn't see my fumbling on the last one.
Yeah.
So watch lead. I'm going to post up for this just in case, and if you decide we'll continue on, then we will.
Yeah, so no, there's a consensus to collect this coral and then possibly the rock, and then we might be out of time at that point.
Okay. I'll try to do it as safely but quickly as we can so that we can try to make it up farther.
Thank you.
We'll be as efficient as we can.
Hi guys, if you're going to collect this Chrysogorgid, it's a really weird Iridogorgia, and I would suggest if you can to grab. I know you don't want to take too much, so we can't go down and get the place where the secondary branching has occurred, but it is important to get a couple of turns in the coil, because that's the measurement that has to be made to determine who the thing is.
Roger that, Les. So I don't know if you heard that, pilot, but
while I get set up, yeah, I did hear it. I need a couple coils from the main stock. Is that what I understand? Watch lead.
Yes, that's correct.
All right. Roger. Hopefully this collection goes better than the last.
Nav, co-pilot.
Good co-pilot?
I believe we have a sample already in the port bio.
Yes, we have, port outer
Okay.
biosample.
So I'm going to extend the starboard pin.
Okay, and retract the port.
So
that one's already done.
we are currently 1,156 meters, and we're just setting up to collect our second biological sample. This is an unusual-looking Chrysogorgid, and it's going to be in the genus Iridogorgia. And Dr. Les Watling at the University of Hawaii has been following us from the Exploration Command Center at the University of Hawaii, and he works on this group and recognized immediately that this is very unusual.
Let's see if we can get
so we collect. We're going to collect a
possible, so I can
sample.
Make sure I get a couple of them.
Permission to zoom in on to focus.
Go ahead.
Yeah, you can even leave it partially zoomed if possible. It's so dark.
You could ask the
Yeah,
box
I'm adjusting.
first too. I can tilt the lights down.
Back a little bit.
There you go.
There you go. Okay. I think this is going to be the best view.
Okay. You come a little more wide. I want to look at the boxes on Mini Zeus.
On H2?
Yes, please.
I've lost control.
So, watch lead. Watch lead pilot.
Go ahead, pilot.
I'm looking, and I think I see two coral in front of me that are the same type. Do you agree?
I've lost control of my zoom.
Copy.
Yes.
There's one in the foreground with a little thicker stalk, and there's one in the background that's possibly thinner stalk and maybe a little longer.
Yeah, that's correct. Yeah.
Which one do you want? Either?
Yeah, either, whatever is easiest to get.
Okay. All right. I'll go for the one in the foreground, I think.
Yeah, they do want a couple of coils.
Yeah, you're also hitting the rock here as you go out, so it's pushing me off. Just so you know, I can get my other toe on the edge here, so that won't happen. Let me just realign. Sorry, watch lead.
What am I doing wrong here?
And there's another one of these
So
Atlanticella
make sure you retract both
sponges in the back.
Yeah. Okay, maybe that was it. It was on that little hump on the...
Yeah, exactly.
I'm going to slowly extend. Sure.
You can leave it in for now.
Okay. You want to collect and then come
Yeah.
off? Okay.
Yeah. Well, I don't have to come off. I just need to...
Copy.
just resettling so that the drawers don't collide when... Okay, that looks stable again. Okay, hydraulics spun up.
Clear for craft.
Alright, craft hydraulics on.
You can zoom freely on Mini Zeus again, Roland, if you need to.
I'll follow the arm out.
Let me know what you need, pilot.
Could you tilt the Mini down a little?
The Mini?
Yep. Just so I can line up. There's so many little spirals in there, it's hard to tell which coral is which, but
Going back up.
it might all be actually one. We'll know when I get close. Oh, I'm moving.
Down a little more.
I think I'm moving around. So.
I think you could probably cut that coral down at the point where that secondary branch is coming off. Or you could just about, judging by the size of the claw, you could just about take the whole thing. Yeah.
Uh-oh.
Yeah, go a little closer toward the base. That would be good.
Closer to the base?
Yep, that looks like...
Here?
Yep, video thinks that looks right.
Watch lead here?
Yeah, go. Yep.
Okay.
Looks like we got a couple coils in there.
That's great.
The vehicle just moved on me right as I was closing.
Yeah.
I think you got
Kind
the
of
whole
pushed
thing.
off with the craft a little bit. Yep. Your shoulder up.
Yeah.
You got the root.
Yeah, we'll take it. There you go. I think
Okay.
That'll be all right. That'll be
Open
great
up,
specimen.
Open up the box. Yeah, bottom parts.
And looks like you got your commensal as well there, Les.